In this video, we will discuss about some of the strategies for receptors inhibitions through phosphatase recruitment. So we will discuss how the proximity of the receptors either activate or deactivate the intracellular signals. So in the panel A, it shows the interactions of APC, that is antigen presenting cells, with the immune cells. And if it interacts with this item, it will cause these transcriptional activations. But if this inhibitory ligands interacts with this item, then it will prevent. And how this item gets activated and leads to this transcriptional activation is that the phosphorylation of this item occurs, and it will trigger this transcription activations into the nucleus. But if this inhibitory ligands interacts with these item receptors, then it will release phosphatase that will dephosphorylate this item, and this downstream transcriptions process will also get disrupted. And one of the examples of this is shown in this panel B, where we can see that the FC epsilon RI signaling activations through this IgE, it will leads to the phosphorylations of these intracellular receptors, and through this SYK, and that will trigger this FC gamma ERI signaling. But when this FC receptors interact with this FC gamma R2B, then it will cause this phosphorylation of this HSP1, and this will prevent the phosphorylations of this SYK, or it will cause dephosphorylations of this SYK that will dampen this FC gamma RI signaling. So in order for this to be function effectively, the proximity of this alpha and FC gamma R2B is important. And some of the other alternatives of this is shown in the panel C, where they can use DARP fusions, that is anti-IgE and FC gamma Y. So instead of FC, they use anti-IgE, or FCE or FC gamma Y, which is shown here. And they can also use the bispecific antibodies with FAB and anti-gamma E and this FC gamma R2B, or DARP bispecific, that is bispecific of this, that is anti-gamma FC, anti-FC gamma R2B and anti-IgE. These are some of the other alternatives that can be used in place of this. And sometimes these strategies can also be used to trigger apoptosis or prevent this overactivations of the immune cells, which is called the, I'm not able to recall that exact name, so let us just keep it as overactivations. So how the immune cells get activated, that is B cell receptors get interacts with this antigenic liposomes, that is here the AH2 or peanut allergen antibodies is shown, that it will cause this phosphorylation of this SYK, and it will trigger B cell receptors clustering and signalings, and these things can be inhibited by CD22 ligands. So this CD22 is glycans that is on this stall, and it is artificially introduced, and it can interact with the CD22 receptors. It will cause phosphorylation of this SHP1, and it will cause dampen this B cell signaling, and simultaneously it will also trigger apoptosis of these cells. And instead of this SHP1, the SHP2 can also play the same roles. And here another example is shown, where IgE is cause the downstream phosphorylation activations in the SYK, but when this IgE another antibodies that has the CD33 ligands interact with the CD33 receptors, that will cause the phosphorylation of the SHP1 or SHP2. It will cause dephosphorylation of this SYK that will dampen FC gamma RI signalings. And other examples where it will cause the downstream signaling of this FC gamma R1A is shown in the panel F. So activations is occurs using this TNP, where TNP interact with the CD64, that will cause this FC gamma R1A signalings activations through the intracellular phosphorylation of the SYK. And this is about this example is about the U937 cell lines. But when they wanted to check which of this ligand caused this which of the Siglec caused this dampening of this FC gamma R1A, they will use the glycans that is affinity towards either Siglec 7, Siglec 9 or Siglec 3. So out of these three different combinations, which of them will cause dampening of this Fc gamma R1A will help us to determine the Siglecs that plays a role towards the this dampening of this Fc gamma R1A signalings. In the panel G, the different Siglecs examples are shown how their uh, structures are look like, either in the form of this common glycan structures or in the traditional chemistry. For example, the CD22 ligands and the Siglec 7 ligand, and this one is Siglec 9 ligand, the CD33 ligand. And in the panel H, here another example of downstream signaling of these T cell receptors, where the T cell receptors interact with this T cell receptors interact and will cause, here the dampening of signals is shown using PD1. So once the PD1 is activated, it will cause this SHP2 phosphorylations, and that will downstream these T cell receptors or SHP2 phosphatase will cause this dampening of this T cell signaling, because it will prevent the activations of the T cell receptors. And these things can be inhibited by using preventing this PD1 receptors activations using this RIP1 PD1. And now this PD1 is not activated and the CD45 receptors gets activated, and it will cause how these PD1 receptors. So in this example, it shows that how these PD1 receptors can be prevented from the activations using this ligand, this RIPR PD1. So it will interact with the CD45. It will cause the phosphorylations of the downstream of the CD45 that will dephosphorylate this PD1 dephosphorylations, and thereby this deactivation that is done by the SHP2 phosphorylations will not occur, and it will cause this activated TCR signalings. And some of the other modalities instead of this RIPR PD1 can be also be used to do the same functions, that is CD45 PD1 on CD28 SIPR alpha, CTLA4 and LY49A1 or NKG2A. And this is some of the other modalities that can also be used, such as CD45 or CD79 or PRF and MET or EGFR. This has also shown to deactivate these PD1 receptors or preventing these PD1 receptors for activations. So I hope you understood some of the examples of the strategies that can be used and the importance of the proximity of the receptors towards this either activations or deactivations of the cells or particular signaling pathways. If you need to understand more about their these examples in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.